Hi everyone, I'm Anna Sanch, currently on my third year of the MSci course in Forensic Science with Nottingham Trent University, and today I'll be talking to you about my third year project, which focused on the potential roles of microbial communities in death investigations, and at the end you can ask any questions you may have. The decomposition process of a human cadaver involves different stages, including fresh decay, bloat, putrefaction, formation of adipose hair, and dry decay. And these are influenced by environmental conditions. For example, adipose hair is more prone to be formed in humid environments. Forensic scientists analyze these stages to estimate postmortem interval, analyzing stages such as livor mortis and rigor mortis, and also body fluid purging, while taking into consideration the temperature and humidity of the environment when the body was found. These stages are commonly paired up with the microbiome, since it's a driving force in the process of decomposition. And this is the community of microbes in the decomposing body, which can also give indications on the cause of death by identification of infections acquired inter vitam, and while differentiating postmortem contamination, as will be discussed later on. The methods of microbial analysis most often used are 16S rRNA sequencing and geochip microarray technology, which help characterize the microbial communities present and are crucial for forensic applications. However, there are still challenges in accessing human cadavers and in replication of the decomposition conditions, which can limit the research in this field. Nevertheless, the current research available still has a potential, shows the potential of microbial analysis in forensic investigations, and this research will be discussed throughout this presentation. So, with this project, the main aim was to outline some of the recent advances made in the qualitative analysis of the microbiome, as well as quantification on some of the body sites, such as the gut, by carrying out a literature review. It was also aimed to characterize the most predominant species found in the literature collected, with more focus on their preferred environmental conditions of these species, as well as their ability to degrade protein, in this case in decomposing matter, for carbon cycling. The focus of this research was in literature present in databases, particularly PubMed, Elsevier, and NTU Library OneSearch, using key terms in the search including forensic microbiology, microbiome, or the specific names of the species found in certain studies or experiments. And along with this review, there was also the analysis of some data received from Dr. Daniel Westcott, which is the director of the Anthropology Research Center at West Texas State University. And some of you might know these research centers, commonly referred to as body farms. So the work carried out in this research consisted of sample collections from the body donations received. For this literature review, there was more focus on body donation D43, as you can see on the diagram present in the slide, which was the body of a 97-year-old male. There was collection of samples from the gut, and which were then processed using a microarray geochip, and this analysis through geochip resulted in over 98,000 data points in an Excel spreadsheet. And then I used the filters present in the Excel sheets to narrow down some data points to use for my project, focusing, as I said previously, on the on species that were responsible for carbon cycling, and then I was more specific and chose carbon cycling with protein degradation, and that eliminated a lot of the data points. And then I eliminated out of those remaining data points the ones that the species that weren't present for 50% or more of the collection period. After applying the filters on the Excel sheet containing the geochip data, it was found that all species that were associated with carbon cycling, particularly the degradation of protein material, were all metalloprotease fungi, which depend on metal ions for their activation. And most of the predominant species were either from the phylum Ascomycota or Basidiomycota. It was also observed that most of the species showed a consistent numbers in terms of population over the sample collection period, as it can be shown in the two graphs on the slide. These were two of the most predominant species found in the data, and the graphs were plotted by grouping different strains of the same species and studying their trends over time. With Arthroderma gypsum, we can see that a few strains maintain low populations throughout the experiment, while there was a specific strain that decreased in the last few hours. And by contrast, there was a strain of Neurospora crassa that in the first 2 days of the experiment had a steep decrease in population, while another strain maintained low population. One of the potential applications of the microbiome in forensics is the estimation of the cause of death. And for my project, I focus on a study that demonstrates the use of histology combined with microbiological evidence to try to differentiate between postmortem contamination and intravenous infections. There were two groups of cadavers used, Group A, which showed signs of infection, and Group B with little to no signs of infection. And the data obtained was summarized in tables that are shown on my dissertation or in the article that is a reference below. And it presents specific cases where microbial examination correlated with the historical findings from the research team, linking pathogens like Staphylococcus epidermidis and Aspergillus to conditions such as sepsis and disseminated aspergillosis, respectively. To analyze the estimation of postmortem interval using postmortem microbiology, two studies were considered. Firstly, we have the Colorado State University study again, which used machine learning to predict the postmortem interval with high accuracy for the analysis of the microbial abundance patterns from the 16S rRNA gene. And the models, particularly the ones where random forests were used, showed that skin decomposer microbes had consistent patterns across different climates, which made them more reliable for postmortem interval estimation than soil decomposers. The other study considered also used machine learning methods and included not only random forests, but also boosted algorithms, specifically XGBoost and neural networks, and it was found that XGBoost was the most accurate one. The microbial taxa that were identified that stand out were Veillonella dispar and Proteus for postmortem intervals above 73 hours, Moraxella for 49 to 72 hours, and Streptococcus for around 48 hours. Majority of the organisms identified for D4E3 are part of the phylum Ascomycota or Basidiomycota. These have been shown in active decay, but Ascomycota can also play a part in fresh decay. And in addition to that, these phyla have been observed in postmortem human necrobiome in the bones, which may indicate that they can also participate in later stages of decay. Due to a lack of in-depth resources on the specific species, their roles in decomposition remain unclear, but it was concluded that the majority of these species, as they are usually found in vegetation and soil, they can be associated with the ecosystem in the research center where the experiment was carried out. When it comes to specific roles, it is known that, as discussed previously, that Arthroderma oti can be related to certain human clinical infections. Therefore, they can assist in autopsies, possibly. And Aspergillus clavitus is a fungus that is often associated with endocarditis and allergic alveolitis, and again can play a part in the making conclusions in the cause of death. Considering the literature reviewed, it can be concluded that fungi and other microorganisms play a crucial role in the degradation of organic matter and consequently decomposition, yet their interactions during this process are still not well understood. The Colorado State University study hypothesized that fungi like Yersinia and Candida break down complex organic compounds such as lipids and proteins into simpler compounds such as fatty acids and amino acids, and the resulting products from this reaction are utilized by bacterial communities such as Ablitomonas alkaphylla for further metabolism reactions, which gives more comprehension of the role of these organisms in the carbon cycling process. But despite these promising results, the studies are still limited by the small sample sizes, for example, and in further studies, one of the potential recommendations for forensic applications would be a comprehensive database that includes the microbial interactions that are still known and the functions that they are known to have in the decomposition process. Going back to the study on estimation of cause of death, it should be noted that the microbial growth and edification of species are not sufficient without the quantitative analysis of the vital microbial loads when it comes to this type of investigation. And sometimes the results can be affected by antibiotic therapy that was administered prior to death. However, other studies suggest that despite this prior treatment, it can be possible to isolate pathogens that are potentially lethal. Both of the studies assessing the prediction of the postmortem interval showed accurate predictions, particularly the second study considered where there was an accurate estimation of postmortem interval within 3 calendar days, which is crucial to establish important timelines for the criminal investigations. Nevertheless, there were still some prediction errors in these studies, which could have potentially been due to intrinsic factors such as the body mass index of the person or extrinsic factors like precipitation or scavengers. Overall, it was a challenging process to interpret the microbial data on both the geochip analysis and the literature reviewed, not only because there was a lack of in-depth resources on the most predominant species identified, but because of other factors. Firstly, the external environmental conditions. This can include the fauna and flora present at the location, the humidity and temperature values, which can affect the type of phyla present in the body and consequently will affect the rate of decomposition. The majority of these factors weren't stated on the literature I focused on for my project. And with the geochip data, it is known that the research was performed at the San Marcos Research Center, and to facilitate interpretation, I did some research on the kind of flora and fauna that could be present, and there isn't a lot of information available besides the fact that it is known to contain a lot of vegetation. However, it is known that the collection period for D43 was carried out in August 2015, so it was very straightforward to find information on the humidity and temperature conditions at the time. Other factors that can affect the rate of decomposition are the body position, the cause of death, as discussed previously, and any health issues at time of death, which were not stated in both the geochip data and the articles I reviewed. And another critical factor to consider in microbial data interpretation is the decomposition stage of the bodies at time of collection. There was an article discussed previously by Hyde from 2014 that states that the experiment was carried out in bodies that were between the pre-bloat stage to the end bloat stage or putrefaction, but besides that article, there wasn't a lot of clarification from other literature and the geochip data on the stage where the collection period was carried out, which makes comparisons of microbiomes difficult. All things considered, the correlation between microbial communities and death investigation was strengthened overall in this literature review. There was a vast diversity of microbial species identified across these studies and the further research done to improve interpretation, as well as in the geochip data. However, as it was mentioned on the previous slides, the interpretation was still limited by lack of resources. Therefore, that aim for this project was not fully achieved. However, it should still be noted that for the aspects of death investigation considered, so estimation of postmortem interval and cause of death, there was improvement of research on those areas from the articles reviewed. The accurate estimation of the PMI within 3 calendar days using microbes paired with neural networks, random forests, and XGBoost showed great improvements for criminal investigations. And the use of histology with postmortem microbiology also showed great potential from the study reviewed for forensics. However, these are still ever-evolving subjects that require further studies, as has been discussed throughout this project. Thank you.